So now we begin with another technical topic from that comes in the chapter of nutrition in plant. And here we will talk about that what are the factors that can have an impact on the rate of photosynthesis. And whenever in biology we talk about the rate, so what our idea is that we always have to speak with respect to the time. Like we know it well that for the rate of photosynthesis, the carbon dioxide and the water, they are my reactants, and they would make the glucose and the oxygen gas, so they would be my products. So I have to see that in how many of the time I would be able to convert this CO2 and the water into the glucose and the oxygen gas. So since this is a chemical reaction, so there would be some factors that would have an impact that to what extent or for how much period of time I would be able to carry out this photosynthetic process. So there are both internal and external factors. If I were to talk about the internal factors, so they would be the number of the chloroplasts that I have with me and how much of the water I'm taking toward the leaves of the plant. So they would be acting as my internal factors. And as far as these external factors are concerned, so they since they are available or the plant take them from the external environment, so they would be the external factors with me. And among them, I have got this concentration of the carbon dioxide gas. I have got this light intensity, and then I have this temperature. So all of these they would be acting as an external factors. Although this clicks in our mind that the water and the ions we also take it from the external environment, but before the water and the ions they can start to have an impact on the rate of photosynthesis, it would affect some other, you could say, situations in the plant in terms of the turgidity and the things like that. So among the external factor, we have got this, this, and this. Why the CO2 is important? Because this is one of the raw material for the photosynthetic process. As far as this light intensity has to be bothered about, so we know this is being absorbed by the chlorophyll to break down this water molecule. So this again is an important external factor. And why we have to be considering the temperature? Because the photosynthetic process would involve some enzymes. So since the enzymes are impacted by the temperature, so this again is an important external factor because the plants they are dependent on the external environmental temperature for the rate of photosynthesis. Now, before we try to crack this topic, your idea of the limiting reactant and the excess reactant should be very much clear. Whenever we say that some reactant is limiting, so as our limiting indicates that any of the reactant that would be used up in a chemical reaction first and would have an impact or would determine the extent to which the chemical reaction would proceed. So this would be the limiting reactant. And whenever we talk about the excess reactant, so it would be present in more amount and it would not be completely used up in a chemical reaction and cannot determine the extent to which the chemical reaction can proceed on. So this would be my excess reactant. Now, if I were to make this point very clear to you, you have to consider a situation. Like I would give you an example from the daily life and a very kind of a general chemical reaction where I have got the reactant A and B and they are being converted into the product C. Now, if you were to see for the reactant A, I have got total of 5 particles of it, and for the B reactant, I have got total of 8 particles of it. Now, the situation is that A would collide with the B and we would make a product C out of it. So you see that A particle, when they keep on colliding with the B reactant, so ultimately, when 5 of the A would collide with the 5 of the B, so at this point, the chemical reaction would stop. Why? Because A reactants were present in less amount, so they would be used up first in a chemical reaction, and that is why this A is limiting and the B is excess with me, and this is only the A which is determining the rate of a chemical reaction. Now, if I were to quote you another example, so you see that whenever we intend to make a normal chicken patty burger, so we are in the need of 2 buns, and then we also require 1 chicken patty for it, isn't it? So the situation is that what I'm gonna do is that I would take the patties, chicken patties of two only. Like I would not increase the number of the chicken patties beyond two. And what I am supposed to do is that as the time would pass, I would keep on increasing the number of buns. So at this point, you see that this bun is one and the chicken patties they are two. So ultimately, chicken patties are excess, and this is this burger bun. This is the limiting with me. But now the time keeps on passing, and now I increase buns to two. So now you see that I would be able to make one burger out of it. Now I further increase the number of buns to three. Again, I would not be able to make any burger, but when I increase it to four, I would end up making another burger out of it. Now I keep these chicken patties number at two and the buns number I keep on increasing. So you see that beyond four buns, now these two burger buns that we have got, now they would become excess. And in this example, the chicken patties are the one that have determined the rate of reaction, not the chicken bun. So initially, the chicken patties they were limiting, they were the excess, and the bun when we had only one, so this was limiting. But later, when the number of the bun has got increased to this much amount, so my chicken patty would become limiting, and the buns that we have got, so they would become excess. So this is something that you have to keep it in your mind to crack what we have got for the photosynthesis. So if we were to apply the same concept that I have referred to you as of now, so you see that among CO2 and the water and the light that we have got with us, so we would only bother about the carbon dioxide, we would bother about the light and the temperature that can have an impact on the extent to which this rate of photosynthetic process could be carried on. And any of if this is CO2 or the light intensity or the temperature that would become limiting, so they would have an impact on this rate of photosynthesis that we have got. Now, since this investigation or this concept cannot be clearly understood if we do not carry out some experiments. So, since we are about to perform some experiments, so you see that whenever we perform any of the experiment in the biology or any of the subjects, so this would involve total of three fundamental variables. The first variable that we have got with us, so this is the independent variable, and by the independent variable, what we mean is that anything that we would investigate or that we would change in an experiment, so this would be my independent. Whenever I talk about the dependent variable, so this is something that I would measure during an investigation or during an experimental work. As far as these constant or the standard these variables are concerned, so I have to always keep them constant. I would not allow them to change. Because if I would allow these to change, then I can clearly not see the overall impact of this independent variable. Like if I were to clear your concept of these three variables again from a daily life example, so at one random day, you people have planned to investigate that how come the salt addition can have an impact on the taste of the rice. Now you know that whenever we intend to be cooking the rice, so it would be the amount of the rice that we have, the spices that we are adding, the oil that we are adding, and the time for which we are providing the heat to the dish. So these are all the factors that would have an impact on the rice. But since I just want to see the impact of the salt addition on the making of the rice, so in this investigation, I would keep the amount of rice, the spices, the oil, and the amount of heat constant. And every time when I'm making the dish, so I would keep on adding a different amount of salt. And what is my independent variable here? This is the salt. And what are my dependent variable? That would be that after we have ended up cooking the dish, so I have to take a spoon and then I have to taste the rice at how come the amount of the salt that I have added, they are impacting the taste of the rice. And I have to keep the other things, like as I've said earlier, that the meat and the heat and the spices and the oil that I'm adding. So I have to keep them constant because if I would not keep them constant, then I would not be able to see that how salt alone is impacting the taste of the rice. Okay, so here we begin to be discussing the first external factor that would have an impact on the rate of photosynthesis, and this is the light intensity with me. So if we just look on to word the statement, what this says is that there is the student Haris, and he wants to investigate at how come the light intensity can have an impact on the rate of photosynthesis. So what Haris needs to change would be the light intensity, so it would be my independent variable. And what he is measuring out of the investigation is the bubbles of the oxygen gas that would be formed, so it would be the dependent variable. Now, as I have said, that since he is investigating the effect of the light intensity, so he has to decide some of the constant or standardized variables. So we know that apart from the light, this is the carbon dioxide concentration, this is the temperature, and the type of the plant or the species of the plant that he would be involving in the investigation. So he have to keep these as constant or standardized. If he would not keep them controlled, or he would not be able to set them at an exact amount, then we alone would not be able to see the impact of the light intensity. Okay, so we have to see that how he will be able to be setting up the experiment in order to see the impact of this. So what he is going to do is that he would take a lamp, okay, and then he would take this beaker. In the beaker, he would take this aquatic plant, and then on the above of the aquatic plant, he would place this inverted funnel, and then he would be placing this test tube on the top of it. And now he would count up that how much bubbles of the oxygen gas they are being produced. So here, since this light intensity is my independent variable, so either I have to move the lamp toward the plant or I have to move it away from the plant. So this is how the light intensity would be changed. So this distance of the lamp from this plant is our independent variable because by changing the distance, we are changing the independent variable. The type of the plant, okay, and the size of the plant, this is my constant variable. The temperature, I'm maintaining it at 25 degrees centigrade. So this again is my controlled or the constant variable. Now, since I also have to be controlling the amount of the carbon dioxide gas, so what we do is we would add up the hydrogen carbonate into it, and that would be keeping the concentration of the carbon dioxide constant. So this is light is independent, the temperature, the type of the aquatic plant, okay, and the concentration of the carbon dioxide, they are the controlled variables, standardized variables. And the bubbles of the oxygen gas that I am counting up now, so they are my dependent variable because this is something I am measuring. Or by simply looking on toward at how many bubbles of the oxygen gas they are produced in a given period of time. So this would give me an idea that how come the light intensity is having an impact on this. If I were to take this lamp far away from the plant, so less of the bubbles would be produced in more of the time. So the rate of the photosynthesis would be slow. If I would move this lamp toward the speaker, light intensity would increase over time. What I would expect is that more bubbles of the oxygen gas would be produced in less period of time, so the rate of photosynthesis would be greater. Now the situation is that from the very beginning of the chemical from the very beginning of this experimentation, I would keep the temperature constant at 25 degrees centigrade. I would keep the concentration of the carbon dioxide at 0.04%, and the light intensity that I have to change, so we would start changing the light intensity from the very minimum value to the very maximum value. As I told you a while ago, that if I take the chicken patties at as two from the very beginning of making the burgers, and with time I'm increasing the number of the buns that we had with us. So you see that initially when I would have the two chicken patties, so they would be excess and the bun would be limiting. But later, when I keep on increasing the number of the buns that I have with me for making the burger, so later the buns they would become excess and the chicken patties they would become limiting. So the same situation would happen in a while, that initially the temperature and the carbon dioxide concentration they would be acting as an excess. Later they would become limiting. Initially when we would be having the light intensity at a very minimum value, so this would be acting as a limiting, but later when we increase to a maximum, you could say values, it would become excess. So this is what I have said at above, that the temperature and the carbon dioxide concentration initially they would be acting as an excess reactant, the light intensity would be acting as a limiting. But later in the experimentation, the temperature and the carbon dioxide gas they would become limiting and the light intensity would become excess. Now you need to have a grip on the graph. So since in this investigation my light intensity was independent variable, so the independent variable it always comes on toward the x-axis, and we were determining the rate of photosynthesis in terms of the number of the bubbles of the oxygen gas released. So since we are counting the bubbles of the oxygen gas, so they are my dependent variable, and the dependent variable they always come on toward the x-axis. Now you see that this light intensity which we have got on toward the x-axis, so we are increasing it from the very minimum value and then we are taking it to the very max value. Okay, so you see that in as the light intensity is increasing, so the rate of photosynthesis is increasing, but beyond this point, the rate of photosynthesis does not increase, but rather than it becomes constant, even though at all the point on this graph, I have got a very maximum value of the light intensity, but the rate of the photosynthesis is not increasing. And we have to see that what is acting as a limiting and excess in this part of the graph and what is acting as a limiting and excess in this part of the graph. So the trick is that since the light intensity initially would be very less and the temperature was 25 and the carbon dioxide concentration was 0.04, so in this part of the graph, apart from the light intensity, the temperature and the carbon dioxide gas that I have got with me, so they would be acting as an excess here, and the light intensity that I have got labeled here on the x-axis, so this would be acting as a limiting, and the temperature and the carbon dioxide they would be acting as an excess. But the time when we make this graph horizontal, like where this is becoming horizontal, so from this point onward, you see, I have attained a very maximum value of the light intensity. So here the light intensity would become excess now, and the temperature and the carbon dioxide concentration now they would become limiting. Now, since we are done with understanding the impact of the light intensity on the rate of photosynthesis, now we have to look on toward the effect of the carbon dioxide concentration on the rate of photosynthesis. So this carbon dioxide concentration, I have to change it. So this is my independent variable. The other thing that I have to be bothering about the external factors of the photosynthesis that would be light and temperature. So I have to keep them constant. The type and the species of the plant I have to also keep it as constant. So we see that how we have to be setting up this experiment where I am supposed to be changing only the concentration of the carbon dioxide. I would take a lamp and the lamp I would keep it at a particular distance from the beaker that have a plant with me. So in this way the light would be kept constant. I would be carrying this investigation at the room temperature, so the temperature would become constant in that way. But the situation is that the time the lamp would be emitting the light rays out of it, so they can also bring some of the heat with it. So how I get rid of it is that I would take a glass tank, and this glass tank I would fill it up with the water. So that would try to eliminate any of the excess heat that could be generated by this lamp source. Now, the independent variable here is the carbon dioxide concentration. So what I am going to do is that I would try to be changing the percentage that I have got for the sodium hydrogen carbonate solution. I would start from the very minimum percentage and then I would keep on increasing the percentage of the sodium hydrogen carbonate solution because this sodium hydrogen carbonate solution is the source of carbon dioxide. So if I increase the percentage of this solution, then ultimately the carbon dioxide concentration would keep on increasing. I would count the bubbles of the oxygen gas being produced. So this would be my dependent variable. Now we have to see that how does the graph looks for it. So you see that my independent variable is the carbon dioxide concentration. So this is coming on toward the x-axis. The rate of the photosynthesis in terms of the bubbles of the oxygen gas being produced. So this is my dependent variable, which I am measuring. So it is coming on toward the y-axis. What the graph is indicating that the time you have started to increase the concentration of the carbon dioxide, the rate of the photosynthesis is increasing, but beyond this point, the rate of the photosynthesis become constant. So the thing clicks in our mind is that why do at these points the graph is not going upward? Because the reason is that at these part of the graph, the carbon dioxide gas concentration that we have got is in excess, but the temperature and the light intensity they would become limiting. Okay, so again, the idea is the trick is that any of the independent variable that you are given over here, so in this part of the graph, the carbon dioxide is limiting, the temperature and the light intensity would be excess because initially at the very beginning of the investigation, carbon dioxide I have minimum, like it would be less amount. So here we have CO2 being limiting and here the CO2 being excess. Here the light intensity and the temperature that we have got, they would be excess, but in this part of the graph, the temperature and the light intensity, they would become limiting. Now moving onward, we cannot neglect the effect of the temperature on rate of photosynthesis. So again, my independent variable here is the temperature that I have to change. My dependent variable would again be the bubbles of the oxygen gas released. And among the constant variables, we have to think of the other external factor apart from the temperature. And we know that is the carbon dioxide and the light intensity. So they would be acting as my constant variable. Now, how we set up this experiment, this would be the same as we have got over here, but the idea would be that I would uh, make the experiment same way, but here I would bring some thermostatically controlled water bath and every time when we would repeat this investigation, so we would be increasing the temperature for that. So now we again have to look at how come the graph is going to look like. So here you see my temperature is the independent variable, the rate of photosynthesis in terms of the bubbles of the oxygen gas. Now they are my dependent variable, so they are coming on toward the y-axis. What I see is that as the temperature is increasing, the rate of the photosynthesis is increasing, isn't it? And at this point, the rate of the photosynthesis is max. But beyond this point, the graph is going downward. The reason is, that as I've said earlier, that uh, photosynthetic process is under the influence of the enzymes. So you know that when we increase the temperature to a very maximum value, then ultimately the enzymes they would start to get denatured. And as a result of that, at this point, the enzymes that we've got in the plants they would be completely denatured, and the rate of the photosynthesis would stop here. Now here comes the thing from your exam point of view, and over there what the examiner do, they give you multiple graphs. Now we have got total of three experiments being performed here. For the first experiment, what I'm doing is not for the first, but for the three of the experiment, what we would be doing that the light intensity we would be increasing for from the minimum to the maximum point. And for the experiment one, the temperature I would keep it constant at 20 degrees centigrade, and the carbon dioxide concentration I would keep it at 0.04%. So what I see is that as the light intensity is increasing, so here rate of photosynthesis increases, but later you see that this line becomes horizontal. This means that the rate of photosynthesis does not increase beyond this point. The reason is that at this point we have got the light intensity as maximum, but the temperature and the carbon dioxide concentration we kept them constant, so ultimately they would become limiting here. So this means that if I were required to be further increasing the rate of photosynthesis from this point onward, then I either have to increase the temperature or I have to increase the carbon dioxide concentration. So when I would repeat this experiment for the second time, so what I'm doing is that temperature I am increasing, but the carbon dioxide concentration I am again keeping it at 0.04%. So now you see that in the second experiment, the rate of the photosynthesis becomes more greater than the first experiment, but again you see that the graph is becoming horizontal here. So the idea is that here the temperature we have got it at max. The light intensity for this part of the curve we have got it at max. So this means that from this point onward. So my carbon dioxide concentration would only be acting as a limiting reactant, and the temperature and the light intensity they would become excess. Now I plan to see, or I plan to basically want that I further want to increase the rate of photosynthesis beyond this point. So I now have to increase this carbon dioxide concentration. So for the third experiment, the temperature I'm keeping it at max. The carbon dioxide concentration, I take it to the max. The light intensity, I keep it to the max. So the rate of the photosynthesis for the experiment 3 goes even beyond the experiment 2 and experiment 1. But again, you see the situation is at this point, the graph again becomes horizontal. But our idea was that this line would keep on increasing because we are not having any of the limiting now for the third experiment because light intensity is max, temperature is max, CO2 is max. Then why do the graph becomes horizontal? Again, the idea is that in the plants there are some particular or some definite number of enzymes. So no matter if I keep on increasing the amount of the carbon dioxide gas, so there is an extent to which the enzymes would be able to form the enzyme substrate complexes. So we say that at this point, the enzymes they have become fully saturated with the substrate. So ultimately, the rate of photosynthesis max can be reached till here and can't go beyond this. So I hope you would be clear with understanding that how come the external factors they have an impact on the rate of photosynthesis.